Hey, welcome back. Our next unit here deals with diffusion. So starting off, concentration is a measure of how much solute there is per an amount of solvent. So a quick recap of those terms. We saw them very briefly in Unit 1. Solute is dissolved by a solvent, and together they make a solution. So for example, sugar, the solute, is dissolved by water, the solvent, to make a sugar-water solution. You might call it lemonade. In science, we often need a way to compare the concentrations of different solutions. So we have these terms, and of course these terms are going to look very much similar. That's science for you. But make sure you just take a look at them and understand the difference between these terms. So the first one is hypotonic, and this refers to a solution that's less concentrated. Think of hypothermia. Your temperature is lower than it should be, so hypo means less concentrated. We also have hypertonic, which refers to a more concentrated solution. Maybe you have a younger sibling at home who is always way too hyper, way too much energy, way over the top. So hypertonic is a more concentrated solution. And finally, isotonic refers to a solution or two solutions of equal concentration. So using those terms here, the image below, we have four different solutions. A is hypotonic to solution B. See how it's lower concentration than B? B is isotonic to solution C, and then solution D here is hypotonic to all the rest. So these are just comparative terms. Think of the word taller or shorter. Is it possible for someone to be both taller and shorter at the same time? Yeah, absolutely. I can be standing next to someone who's shorter than I am on my right, and someone who's taller than I am on my left. So I'm both shorter and taller at the same time. And the same is true of these terms. They're just kind of descriptors. You can be both hypotonic and hypertonic at the same time. Solution B and C are hypotonic to B, but hypertonic to A, and isotonic to each other. So, yes, it is possible to be both at the same time. All we're doing is using a comparative term. This one is more concentrated than that one. That means it's a hypertonic solution. B is hypertonic to A. So just comparative terms, what we're using here. So diffusion is the natural tendency of particles to spread out from an area of high concentration to an area of low concentration. So what we're watching here is a sugar molecule being dropped into water. And the reason why we don't see that white granular sugar anymore when we put it into water is because it dissolves. Dissolving is that sugar molecule coming apart and it's spreading out evenly in the water. And that's what diffusion is. We see that sugar molecule spreading apart, diffusing from an area of high concentration. See that high concentration in a second here. So here's an area of high concentration, lots of sugar molecules, but they spread out to areas of low concentration. That is diffusion. This does not require energy. It is a natural process. It happens without any energy. Now, because we say particles are moving from high concentration to low concentration, we oftentimes use the term down the concentration gradient. So from high down to low. It's a natural process down the concentration gradient. And we'll use that term, down the concentration gradient, quite often in the next couple of chapters here. So things move from high to low concentration, and again, that's a natural process. Next up, we talk about selective permeability. So selective permeability means that you have a barrier, got this blue wall here, and some items can pass through and others can't. Some items can diffuse straight through, other items are not able to. A lot of times it's due to size. So an example of selective permeability is a window screen. Probably have several of them in your house right now. So a window screen has small enough holes that Air can pass through and get a nice cool breeze on a hot summer day, but it's too small for something larger, let's say like a bug, to pass through. So that window screen is selectively permeable. It allows air to permeate. It does not allow larger substances like a bug to permeate. And that's what we see here. This blue membrane here, this blue barrier, is selectively permeable. It's allowing the red dots to pass through. They're small enough, but the green and yellow dots are not small enough. They cannot pass through. That is selective permeability. All right, so why are we talking about all this? Well, back to the cell membrane. This whole unit is about the cell membrane, and as it turns out, the cell membrane is selectively permeable. One of the most important jobs that the cell membrane has is to regulate what's able to enter the cell and what's able to leave the cell. So being selective is key. It's very important that the cell gets the right things in, keeps the wrong things out, lets the right things leave, and keeps the right things in. So selective permeability is really important for our cell membranes. So particles that can permeate or travel right through the membrane are generally small, non-charged particles. Water, I've got an asterisk there, we're going to cover that in the next page. Oxygen, carbon dioxide, nitrogen, glycerol, these are all particles that are really important for the cell, and so it's important that they can travel right through the cell membrane with relative ease. They all happen to be small, and they are all uncharged. It's another big thing. Particles that are too big to permeate the cell membrane include glucose and amino acids. Those are maybe not necessarily macromolecules, but they're the monomer units, so they're much larger than something small like a water or carbon dioxide or oxygen. There are other particles that are small enough, perhaps, to pass through the membrane, but they are unable to because they're charged. So remember, those phosphate heads on the outside of the membrane are negatively charged, and the tails in the middle are non-charged. So that kind of creates a barrier where positive and negatively charged ions can't travel right through. Hydrogen ion, for example, is much, much smaller than water. Think about it, water has 2 hydrogens in it and a big old oxygen right in the middle. So the hydrogen ion should be not even a third the size of water. But it's unable to pass through the membrane because it has a positive charge. So charged ions and large particles generally cannot just diffuse straight through the membrane. It is selectively permeable against them. All right, on that last slide, we saw that water had an asterisk. Technically, water does not do a great job of just going straight through the membrane. You guys remember, those polar heads love water. That's why they point towards the water on both the top and the bottom of the bilayer. But those tails, those fatty acid tails, they don't love water so much. They are hydrophobic. They fear water. They don't mix with water. So I know this image has some water molecules kind of squeaking through, but that's not so common. It doesn't happen very often that water just goes straight through the membrane. Instead, most cell membranes have a protein that allows water to just go straight in and out. Here's that protein. So see, it's just as packed in that protein on the inside and the outside of the cell and the middle of that protein. And that protein is called an aquaporon. It's even colored aqua here. So aquaporons are integral proteins that allow water to diffuse freely into and out of the water. And that'll be a really important concept tomorrow. So to recap what we looked at today, diffusion is the natural movement of particles from a high concentration to low concentration. And where's this going with what we're learning? Well, the cell membrane allows some particles to travel straight through, but it protects us from other molecules. Other molecules cannot pass straight through. And this is known as selective permeability. All right, well, thanks for tuning in, and we'll see you guys right back here tomorrow.